Hello dear students, welcome to the video, welcome to the discussion of AIATS Plus Phase 2 Mock Test 9, where we discuss the questions in which most of you tend to make mistakes. So, as our ritual has been over the course of our discussion, that we take up topics from these particular chapters. So, they are biomolecules, excretory product and their elimination, locomotion and movement, and these three chapters pertains to class 11th, whereas human health and disease and evolution pertains to class 12th. And I am Snehal, your zoology faculty at Aakash. So let us begin the discussion without wasting much time. So the first question that we have today is question number 136. And it states, choose the incorrect statement with respect to human axial skeleton. So dear students, we know that the human skeletal system is divided into the axial skeleton and the appendicular skeleton. And as I look at the question, I can see that we are particularly talking about ribs. So let us quickly discuss the concept of ribs to deal with this question. So ribs are basically flat bones which constitute our thoracic cage. So basically we have 12 pairs of ribs and they constitute the thoracic cage, as you can see in this diagram. So this diagram represents our thoracic cage. So coming back to the structure of bones, if we say that they are flat, thin bones, flat and thin bones, and their head they are bicephalic, which means that their head will have surfaces which act as the articulation site. So basically bicephalic means, bi means two and cephalic means head. So ribs are said to have two heads on their dorsal side which help them in articulation. So if we imagine that we have our thoracic vertebrae like this, so our ribs will articulate like this. We can see that we have two ends which are articulating. Just imagine that this is your thoracic ribs, and in between you have your thoracic vertebrae, not ribs, and this is your rib, and its dorsal side will contain the surface for articulation, which we are referring to as the bicephalic head. Now, how they are attached to form our thoracic cage is the basis that they are given different names. So basically, there are three types of ribs, we say, and the first one is the true rib, as you can see on your screen in the diagram. So basically ribs are attached ventrally to the sternum, as we can see in our diagram as well, that we have the sternum, and to the sternum we can see that our ribs are attached with the help of cartilage. All right. Then these, so and on the dorsal side and on the dorsal side, our ribs are attached to the vertebral column. So basically, if the ribs are said to have these two attachments, like they are ventrally and dorsally attached to the respective structures, they are said to be the true ribs. And in the figure we can see that we have 7 pairs of true ribs. So we have 7 pairs of true ribs out of the 12 pairs. All right. Now coming, and they are also said to be the vertebrosternal ribs. They are also said to be vertebrosternal ribs. Now we can see that there is another labeling, and it has been named as the false ribs. So basically, the 3 pairs of ribs, which are the 8th, 9th and 10th pairs of ribs, they are connected dorsally to the vertebral column, but ventrally they are not connected to the sternum. Instead, they are connected to the 7th rib with the help of a cartilage, which is known as the hyaline cartilage. So we can see in the figure as well that the 8th, 9th and 10th pairs of ribs are not attached to the sternum directly, rather they are attached to the 7th rib with the help of the hyaline cartilage. So basically we are classifying the different types of ribs on the basis of the type of attachment that they show. And based on these, questions are often asked that how many numbers of false ribs do we have? So the answer will be that we have 3 pairs and 6 false ribs. Similarly, we can see that there is another labeling, and they are said to be the floating ribs. So floating ribs are basically classified as such because they are not attached ventrally with any of the structures. Their ventral side is free, we would say, and they are attached to the dorsal vertebral column only, and they are not attached to any structure on the ventral side, and therefore they are said to be the floating ribs. And it is the 11th and 12th pair of ribs which are known as the floating ribs. So this was the basic about the ribs. So let us now go back to our question to figure out our right answer. So our first statement states that each rib has 2 articular surfaces on its dorsal end that we talked about, and is hence called bicephalic. So this is absolutely true, as we already discussed, that it attaches to the thoracic vertebrae with the help of these bicephalic ends, and therefore option 1 cannot be the right answer. Moving to option 2, which states vertebrochondral ribs are 7th, 8th and 9th pairs. So dear students, as we discussed that we have 3 pairs of ribs which are the false ribs. So false ribs are known as the vertebrochondral ribs. So these are known as the vertebrochondral ribs. And in the question it has been stated that the 7th rib is known as the vertebrochondral rib, which is incorrect, as it is the vertebrosternal rib, as we already discussed, and therefore option 2 is the answer that we are looking for. But let us read the other options as well. So the third option states all ribs are attached dorsally to the thoracic vertebrae, which is true. Then we have the fourth option, which states floating ribs are not attached ventrally to sternum. This is again true, as we already discussed, that they are not attached ventrally to the sternum. So this we can conclude that here the answer that we are looking for is option 2, and it was a simple question. We just need to keep the facts on our fingertips to solve such questions, as all the information is given in the NCERT as it is. Now moving on to our next question, and it states question 137, that malignant tumors are characterized by. So by looking at the question, we can figure out that here we are talking about cancer. So let us quickly discuss the important concepts that we need to remember when it comes to the NEET exam with respect to cancer. So basically in our cell, there is a cell cycle which is operating to keep the division of the cells in check. So basically a cell grows in size, a cell is dying over the course of time, or there is different changes that are occurring within a cell. All of these are regulated by a cell cycle which operates, and there are different checkpoints within that cell cycle which make sure that when the cell cycle will be functional, or when the cell cycle has to stop, or the cell division has to stop. So basically in normal cells we have these cell cycles which are operating properly. But if there is any by chance or via various sources, I would say, there is any disturbance in this cell cycle, then it can give rise to a condition which is known as the cancer or the tumor formation, which is characterized by uncontrolled cell growth. So basically in cancer there is tumor formation. And why this tumor is formed? Because the cell cycle which was operating normally in the normal cell has undergone some sort of disruption, and now the cell growth is in excess, or there is continuous divisions which are taking place, and the cell will grow in size abnormally and give rise to the formation of tumors. Now further, these tumors can be of two types, which are known as the benign tumors and the malignant tumors. Now we will discuss these two different types of tumors, but let us first discuss what is the common characteristics which is seen in a tumor. So basically a tumor cell is said to have undergone neoplastic transformation. Neo means new. So basically now the tumor that has formed has characteristics which is not like the normal cell. So we say that it has undergone neoplastic transformation. What this tumor does is that this tumor will starve the nearby cells, and it will kill the nearby cells or affect the nearby normal cells. It is very obvious that since rapid divisions are taking place in this tumor cells, and therefore it will require more energy than the normal cells, and therefore they starve the other cells, nearby cells, for nutrient, and they grow in size. Now what is the cause of this disruption in cell cycle. So there can be various causes, like there are mutagens which can give rise to mutations which are changes in the nucleotide sequences or nucleotide bases. So basically these mutagens can induce mutation in the normal cell and can give rise to abnormal conditions. So what are the source of these mutagens? Can you please tell me in the comment section? The information about the different types of mutagens is given in the NCERT in a subtle way. So I would expect answers from you in the comment section. So basically mutagens lead to or induce mutations that disrupt the cell cycles. So in normal cells we have these proto-oncogenes which are functioning normally to keep the cell cycle in check. So what mutagens does? The mutagen will disrupt or affect this proto-oncogenes and convert it into an oncogene. So this proto-oncogene which was present in a normal cell gets converted into an oncogene, and thus this oncogene further leads to the neoplastic transformation that we see that give rise to tumors. Like in the figure we can see that our first figure represents a normal cell division. So in the normal cell division we can see that we have a tumor suppressor gene. So the name is very much obvious in itself that this gene is working towards suppressing tumor formation, and one of the examples of tumor suppressor gene is P53. So we have a normal tumor suppressor gene and then we have a normal proto-oncogene. So both of these are regulating or forming proteins which are running the cell cycles under control. So we have our normal cells here, whereas in a malignant cell division or in an uncontrolled cell division which we see in the tumor cells, we can see that we have a mutated tumor suppressor gene or the mutated proto-oncogene, which is also known as the oncogene, and we can see that it has led to various protein formations which are leading to an abnormal cell cycle, and thus we can see we have our cancer cell formation. Further we can see in the figure here that we have a normal cell in the first figure, and we can see our cell where there is a nucleus, and we can see the cells that have distinct or proper boundaries, whereas in a cancer cell we can see that they are morphologically very distorted and there is no distinct boundary to define those cancer cells. So this is one of the major differences between a normal cell and a cancer cell. Like if I give an example of a process or a phenomena which is the contact inhibition. So basically in a normal cell what happens is that when the cells come in contact during their division, then they will not further divide if they come close to each other. So normal cells are said to show or exhibit contact inhibition. So basically when they are dividing, they will stop dividing if they come close to each other. Whereas in a cancer cell, we can see that these cells are continuously dividing, and in fact they are overlapping each other. So we say that normal cell forms a monolayer, whereas a monolayer cannot be seen in a tumor cell because they are an aggregation or a lump of many cells and they lack the property of contact inhibition. So this is one of the important differences between a normal cell and a tumor cell. Then further we can see that here we have a normal cell and we can see a precancerous cell where a cell has lost its property of contact inhibition and now it is dividing in an uncontrolled manner and giving rise to, and it can give rise to a tumor cell. Now coming back to our different types of tumors that we discussed early is the malignant and the benign tumor. So malignant, as the name suggests itself, is evil. So we say that malignant tumors are evil or dangerous because they exhibit a property which is known as the metastasis. So basically what happens is that the cells slough off from the malignant tumors and they reach the distant sites present in our body via blood, and then wherever they reach they lodge themselves there and start forming a new tumor there. As we can see in the diagram here that we have a malignant tumor and we can see that the cells from this tumor is moving out and it will further go to different sites, and wherever they lodge they will start forming a different tumor there. So this is known as the property of metastasis, which is exhibited only by the malignant tumor. All right. Whereas if we see the benign tumor, we can see that the benign tumor has an intact boundary, or it is like a capsule, we can say in a simple word, and the cells present in these tumors are not moving out or not slouching off, and therefore they do not spread to different sites, and therefore they are considered to be less dangerous as compared to the malignant tumors. It is very obvious that since they are intact to a particular location, their treatment of the malignant tumors is comparatively more easier as compared to the malignant. The treatment of the benign tumor is easier as compared to the malignant tumor. So we go back to our table now. So the malignant tumors will exhibit the property of metastasis, whereas benign tumors do not show this property, whereas contact inhibition, as we already saw, will be absent in both of them. Both of them will be said to have undergone neoplastic transformation. So we say that neoplastic transformation has occurred in both of them. Then we have another way to say that malignant tumors are more dangerous, as we say that they are more invasive in nature. They are invasive, whereas benign tumors are non-invasive. Now both these tumors will starve their nearby cells and they will compete for nutrients. So both of them will starve or affect their nearby cells. So this was the basic differences between the different types of tumors. So now we go back to our question. And our question demands that we need to choose the characteristics of a malignant tumor. So A is given to be presence of contact inhibition. So we saw that contact inhibition is exhibited by normal cells and not malignant cells. Therefore, A is incorrect. B states property named metastasis, as we already discussed and saw in the diagram as well, that malignant tumors exhibit the property of metastasis. So we'll include B in our answer. Then lastly, we have rapid and uncontrolled growth. So we already discussed at length that tumors are characterized by rapid and uncontrolled mitotic growth, and therefore C will also be included in our answer. So we can see very clearly that we have B and C, which should be the answer, and our second option states the same. Therefore the right answer will be option 2. So this was the basic about the different types of tumors. Now I can see that students have joined in the comment section. A very warm welcome to all of you. So now let us go back to, now let us move ahead to our next question. So the next question that I see is question 138, and it states, which of the following is correct with respect to enzyme inhibition? Alright, so basically this question pertains to the chapter of biomolecules, and I have repeatedly mentioned in my various sessions that biomolecule is one such chapter from which most of the questions are asked in the NEET exam. So let us try to solve this question. We can see that in the question they are talking about the different types of inhibition. As I can see the word competitive and non-competitive. So let us quickly recall the concept which is related to the enzyme and enzyme action. So basically we know that enzymes are, most of the enzymes are proteins which are known as the biocatalyst, and enzymes function to accelerate the rate of reaction that is occurring in our body. Alright, and enzymes function by lowering the activation energy that is required for the formation of product from a substrate. And to define it properly, activation energy is said to be the difference in the energy of the substrate and the transition state. So for achieving or for the formation of product, the substrate should be able to cross this activation energy barrier, and after that the product will be formed. So our question was basically about the types of inhibition. So whenever enzymes function, the mechanism of enzyme works by formation of enzyme substrate complex. So basically we will have our enzyme and a substrate will bind to the active side of the enzyme and induce conformational changes. There will be breaking of new bonds or formation, there will be breaking of old bonds, formation of new bonds, and there will be different altered structural states which are also known as the transition state, and finally our enzyme binds to the substrates and forms the enzyme substrate complex. After the formation of enzyme substrate complex, there is the formation of enzyme product complex, and eventually the product is released and the enzyme is free again to be used in the reactions further. So it was a basic, that is the basic layout of how enzyme function to carry out a reaction. But here in the question we are talking about inhibition. So inhibition in general means to stop something, to inhibit or to slow down the activity of anything. So we know that the function of enzymes can be controlled or can get affected via various factors such as temperature, pH levels, or the presence of cofactors. Also they can be affected by the presence of inhibitors. So I can see that Srinath is asking that potential energy of transition state is more than the potential energy of the substrate. So Srinath, you are right that the potential energy of the transition state will be more than the substrate, and we will see that in our questions further in the figure. So as we already discussed the basic layout of the formation of product from an enzyme, formation of a product from the formation of enzyme substrate complex. So here now we were talking about the presence of inhibitors, as to how the presence of inhibitors can affect the enzymatic activity. So as we already discussed that we will have an enzyme and a substrate will bind to it. So what happens in competitive inhibition is that we have our enzyme, as we can see in our on the image that we have on our screen, we have our enzyme and we have our substrate. But there is also an inhibitor present here which somewhat resembles the substrate. So basically our inhibitor resembles the substrate structurally. It will resemble the substrate structurally, and as we already discussed that the substrate binds to the enzyme. So here the normal tendency of the substrate is to bind the enzyme, but what happens is that since there is a structural similarity between the substrate and the inhibitor will bind to the active site of the enzyme, and it will prevent the substrate from binding to the active site. And if the substrate is not binding to the active site, then obviously there will be no reaction, or the rate of reaction will decrease. So in the image we can see that due to structural similarity, the inhibitor got attached to the active site of the enzyme, and instead of the formation of the enzyme substrate complex, there is a formation of enzyme inhibitor complex. So inhibitor binds to the active site and it directly blocks the active site. And since the active site is blocked, no more substrate can bind to it, there will be no formation of enzyme substrate complex, and therefore there will be no reaction. Now, one of the important features, I would say, of the competitive inhibition is that it can be overcome by adding more number of substrates. So basically what happens that if there are more number of substrates as compared to the inhibitor, the substrate will be able to bind to the active site, or there will be formation of more number of enzyme substrate complex as compared to the enzyme inhibitor complex, and therefore the reaction can be, the rate of reaction can be overcome with increasing the concentration of substrate. The simple logic is substrates के molecules अगर ज़्यादा है inhibitor से, so there will be a tendency that the substrate will bind to the enzyme, and the number of enzyme substrate complex formed will be more as compared to the enzyme inhibitor complex formed, and therefore the rate of reaction can be regained again to its normal rate. So this is one of the important hallmark of competitive inhibition, and one of the examples that is given in the NCERT for competitive inhibition is the reaction of succinic dehydrogenase. So succinic dehydrogenase binds with the substrate which is known as the succinate. But there can be a competitive inhibition when malonate is also present, because there is a structural similarity between succinate and malonate. So we say that the inhibition of succinic dehydrogenase by malonate exhibits competitive inhibition, but it can be overcome if we increase the substrate of concentration, which is succinic here. Alright. So this was the basic of the competitive inhibition. Now we also saw that in our question we had discussed about non-competitive inhibition. So the difference between the competitive inhibition and non-competitive inhibition is that the inhibitor which is present here will not bind to the active site of the inhibitor. As we can see that we have the inhibitor here. So the inhibitor will not directly bind to the active site of the enzyme. Instead, it binds to the allosteric site of the enzyme. The inhibitor will bind to the allosteric site of the enzyme, and it will induce such changes in the active, such conformational changes that there will be a change in the shape of the active site of our enzyme. So होता क्या है कि inhibitor will bind to the allosteric site. Now this will bring about a conformational change in the active site of the enzyme, और जो enzyme का active site है that will undergo a change in shape. For instance, we can see that the shape of our substrate is like a triangle here, but since our enzyme, our inhibitor has got bound to the allosteric site, the active site, the shape of the active site will change from the conical shape to, let's assume, a circular shape, and therefore the fitting of this substrate into the active site will not be proper. So since the substrate will not be able to bind to the active site properly, there will be a decrease in the rate of reaction, as formation of enzyme substrate complex is a mandatory step during the course of a chemical reaction. So this is one of the important differences between competitive inhibition and non-competitive inhibition, that in competitive inhibition the enzyme was, the inhibitor was binding to the active site directly, whereas in non-competitive inhibition the inhibitor is not binding to the active site, instead it is binding to the allosteric site, which is inducing conformational changes that leads to a change in shape of the active site. Alright. Also, by increasing the concentration of substrate, this non-competitive inhibition cannot be elevated. Like we discussed that in competitive inhibition, if we increase the concentration of the substrate, then the effect of the inhibitor can be diluted, but that does not happen when we talk about non-competitive inhibition. Alright. So now we go back to our question, since we have discussed about the different types of inhibition. So our first option states addition of lots of succinate does not reverse the inhibition of succinic dehydrogenase by malonate. So dear students, as we discussed already, that succinate is the substrate, and the reaction of succinate with succinic dehydrogenase in the presence of malonate represents competitive inhibition, and it can be overcome by increasing the concentration of the substrate. Therefore, our option 1 is incorrect. Moving on to our option 2, which states a non-competitive inhibitor binds with the enzyme at the same site where substrate binds. So dear students, we discussed that the substrate will bind to the active site, whereas the non-competitive inhibitor binds to the allosteric site, and therefore option 2 is also incorrect. Moving to our option 3 now, which states inhibitor has to resemble the substrate in non-competitive inhibition. So this is again an incorrect statement because such condition does not exist. We specifically call it competitive only because there is a competition which is existing between the inhibitor and the substrate to bind to the active site, because they are similar in shape or structure, and therefore option 3 is incorrect. Now moving to our last option, which states in competitive inhibition the competition is between substrate and inhibitor to occupy the same active site. So as we already discussed at length, that the substrate will bind to the that the inhibitor will bind to the active site of the enzyme such that the rate of reaction decreases, and therefore the answer here that we are looking for is option 4. So the discussion about non-competitive inhibition is not given in the NCERT, but we have been asked question in our NEET exam in previous year's papers. So it is important that we are thorough with the basics of these two different types of inhibition. We need not go in much detail, but if you know this much, it will be enough to tackle any of the previous year questions. All right. So now we move on to our next question. All right. So our next question is question 144, and it states how many bones given in the box below are not included in axial skeleton. So students, as we already discussed, that our endoskeleton is divided into two principal axes, which is the axial skeleton and the appendicular skeleton. So basically, to deal with such questions, we need to remember the components of the appendicular skeleton and the axial skeleton. So for your ease, we have a chart here that can help you remember the names of the components which are a part of the axial skeleton, or just to make it easy for you, we can remember it by keeping in mind that whatever falls along the axis of the body are categorized as under the axial skeleton. For instance, we have our cranium, we have our sternum, which is characterized or falls under an axis, along an axis, and therefore they are categorized as the axial skeleton. Whichever part which is moving away from the body is categorized under the appendicular skeleton. All right. As we can see in the figure as well, that we have bones along one axis, and it is a part of our axial skeleton. So basically, we have our skull, which is made up of the cranium bones and the facial bones. Then we have the hyoid bone, which is a U-shaped bone that is present at the base of our buccal cavity. Then we have the auditory ossicles, the stapes, incus and malleus. We say that we have 3 pairs of auditory ossicles. Then we have the vertebral column, and then we have the thorax, which is made up of sternum and the ribs. So all these parts constitute the axial skeleton of humans. Now we'll quickly recall the appendicular skeleton, and as we say that, as we discussed, that whichever part which is moving away from the body, like our hands, the upper limbs and the lower limbs, constitute the appendicular skeleton. So we can see we have our girdles. We have our girdles, which is made up of scapula, and clavicle. They are categorized under the appendicular skeleton. Then we have our upper limbs, which are made up of different bones. Then we have our lower limbs, which are categorized as the appendicular skeleton. So the easy way out to remember this is that we have our limbs and girdles, which are categorized as the appendicular skeleton. So now we go back to our question to figure out that which of the given bones are not included in the axial skeleton. So dear students, it is very important that when you read the question, you make sure that you focus on the details of the question. For instance, it is written that we need to choose the bones which are not included in the axial skeleton. All right. So first we have the sternum, and we saw that sternum is a part of our thorax, and sternum is a very much a part of axial skeleton. Therefore, we will not include sternum in our answer. Then we have the vertebra. So vertebra are also a part of the axial skeleton, so we will exclude vertebra. Then we have the femur. So femur, we know that is a bone of it is a bone of our hind limb, and it is also known as the longest bone which is present in the human body, and it is a part of the appendicular skeleton. Then we have the carpals. Carpals are a part of our forelimb, and therefore we will include carpal as well in our answer. Then we have the scapula. Scapula is a part of our pectoral girdle, and therefore we will include scapula as well in our answer. Then we have the ethmoid bone. So ethmoid bone is the unpaired cranial bone, and it will be a part of the axial skeleton, and therefore we will not include ethmoid in our answer. Then we have the maxilla, which is a facial bone, and therefore it will also categorized as the axial skeleton. And thus in total we can see that we have just 3 names which will be categorized as the appendicular skeleton, and we were looking that only in our answer, and our second option states 3. Therefore the right answer will be option 2, and we will straight away eliminate the other options. So basically, dear students, to deal with such questions we need to focus on the lines which are given in the NCERT, as it is very clearly mentioned in the NCERT that the ribs and the cranial and the facial bones constitute the axial skeleton, whereas the limbs and the girdles constitute the appendicular skeleton of the humans. And this is how indirectly you can be asked questions via naming the different types of bones. All right. So this was a tough question. I would categorize it as a tough question, since it is difficult to remember the names of the bones. All right. So now we have our next question, which is question 142. And I can see that it is a diagram based question which is taken from the chapter of biomolecules. And the figure which is given to us represents a diagram where potential energy of the substrate and the product has been depicted. So to deal with this question, I can also see we are talking about the endothermic and exothermic reactions. So let us quickly revise the concept of different types of reactions. So basically, reactions or the chemical reactions can be of 2 types, which are the endothermic reactions and the exothermic reactions. So as the name itself suggests, endothermic reactions involve or require heat so that it can occur. So basically, endo means to supply heat to the system. So heat is required in endothermic reaction, whereas exo means out, and basically in exothermic reactions heat is released from the system. So one of the remarkable differences or the major differences between an endothermic and an exothermic reaction is that endothermic reaction requires heat, whereas exothermic reaction involves release of heat. And we can see this, that the first figure that we have here represents our endothermic reaction. Sorry, it represents our exothermic reaction, because heat is being released from the system, whereas our second figure represents the endothermic reaction, because heat is being supplied to the system. So this was a basic about the endothermic and exothermic reaction. Now if we try to talk in terms of the potential energy of the substrate and the product, then as we can see that heat is being released from the system, then obviously the level of potential energy will be less in the product when we are talking about the exothermic reactions. So this figure has been taken from the NCERT book itself, and since it is in the book itself, in the NCERT book itself, the detailed explanation is not given, but this graph represents an exothermic reaction, because as we talked that heat is released in the exothermic reaction, so the product will have less heat as compared to the substrate, and therefore we can see in our figure that the product is at a lower energy level. Here we can see we have a potential energy marked at the y-axis. So the product is at a lower energy level as compared to our substrate. So basically this diagram is representing an exothermic reaction. Had it been the endothermic reaction, since we talked that in endothermic reaction heat is supplied to the system, therefore if we try to plot the endothermic reaction, then we have our potential energy here and we have our progression of reaction as given in the other figure. So our substrate in this figure our substrate will be at a lower potential energy level as compared to the product, because the product has been formed by absorbing the energy, so it will be at a higher potential energy level when we talk about the endothermic reactions. Also, a student was asking that the potential energy of the transition state is more than the substrate or not. So we can see in the reaction clearly in the graph that the transition state is at a higher potential energy level as compared to the substrate. So be it exothermic reaction or the endothermic reaction, the transition state will always be at a higher potential energy as compared to the substrate. So for better clarity, we can have a look at this figure. So my first figure represents an exothermic reaction. So we have a reactant here and we have the product here at a lower energy level. Whereas in the endothermic reaction, we can see we have our substrate at a lower potential energy level, whereas our product is at a higher energy level. Now, if we talk about the activation, the concept of activation energy, we already discussed that the enzymes act as the biocatalyst and they lower the activation energy which is required for the substrate to form the product. So in the presence of enzyme, we can see that we have, in the presence of enzyme, we have a lower energy barrier that needs to be crossed whereas without the enzyme we have a higher energy barrier for activation energy which needs to be crossed. So basically enzymes act as a biocatalyst by lowering the energy, by lowering the activation energy. So now we go back to our question. And now after the discussion, I hope that we will be able to figure out that the given reaction is representing an endothermic reaction or an exothermic reaction. So we can see that we have our potential energy and the substrate is at a lower energy level as compared to the product. So this is definitely representing an endothermic reaction. Now we see that there are two other labelings which are known as, which has been written as A and B. So we can compare this with our other diagram which is given in the NCERT, which is indicating the activation energy, that here the labeling which represents a higher energy barrier, the higher energy barrier must be the activation energy in the absence of enzyme. The enzyme is absent here, whereas the energy barrier which is lower and represented by A must be the activation energy in the presence of enzyme. So if we are aware of the concepts of the different types of reactions, we can easily deal with this question. Otherwise you might find this question a bit tricky, but the simple trick here is to look at the levels of the substrate and the product to figure out the answer. So now if we go back to our question, our first option states endothermic reaction with activation energy A in the presence of enzyme and B in the absence of enzyme. So dear students, as we discussed that it is an endothermic, this graph is representing an endothermic reaction, which is true, but here A represents the activation energy in the absence of enzyme. Therefore, option 1 will get eliminated. Option 2 states exothermic reaction, so we will straight away eliminate this option. Option 3 now states endothermic reaction with activation energy A in the absence of enzyme, which is absolutely true, and B in the presence of enzyme. So the answer that we are looking for here is option 3rd. Option 4th will also get eliminated, as we see exothermic reaction. We need not even read the full option. So dear students, this was the basic about the concept of different types of reactions, and since we have one of the figures given in the NCERT, it is important that you are aware with the other type of reaction as well, because in the NEET exam such questions can be asked. Alright, so this marks the end of the discussion, and I hope that you enjoy the sessions that we have had so far. All the best for the upcoming NEET exam and hope to see you again. Good luck and thank you.